class seems to have shrunk another order of magnitude. But I guess we'll get started. Before we get started, does anybody have any questions that you would like?
I have a lot of questions about the review, but I assume
Okay, we'll
do
start
that
on
later.
those after this lecture
Yeah.
then. I'm not going to use the whole time to lecture. I want to use some of the time for questions. But I wanted to try and present this paper for you to kind of illustrate the value of being able to think outside the box. I mean, to be able to draw on data not only from your own particular area of study, but from other areas of studies in order to think about or identify new approaches to your particular problems. And this is obviously one of the, if you've had a chance to look at it, one of the more surprising papers that I've seen in a while. It came out in Cell this summer. That's a pretty high prestige journal. It normally indicates that it's undergone extensive vetting and that they would only publish data they felt had been convincingly demonstrated. And the authors clearly went out of their way. Well, they were probably forced to go out of their way to really test every possible outcome and explanation, alternative explanation for what they're looking at. But what they found is quite unusual. So I want to talk a little bit about this because in addition to being an unusual paper, of which there's many, it draws on a number of the methods, techniques, and proteins that we've discussed in this course, including ones very recently. So it kind of gives you a, maybe even if you don't work on things related to multicellular eukaryotes or gene silencing or even viral vectors, it gives you an idea of how sometimes those techniques and those proteins can really change the way you're looking at things. So what we're going to be talking about is this paper where what they essentially did was take two different RDRP proteins. Remember, RDRP are those RNA-dependent DNA polymerases. One came from a plant called Arabidopsis. One came from another plant, rice. And they introduced each of these genes into a mammalian cell line. Now, they don't really tell you why they introduced it, but I'm going to make a guess based on the way the things seem to evolve. This is another, in my mind, another possible case for the things that we discovered by serendipity. In other words, I think they were introducing these two different plant proteins, these two different RDRPs, into mammalian cells in an attempt to control virus infection. Because in plants, we know these RDRPs played two or at least two primary roles. One, they're a defense against certain types of RNA viruses. And two, they are a defense against over-excitation of many types of transposons based on retrovirus ancestors. In those cases, we know from the previous studies that if you mutate the RDRP of plants, they do become more sensitive and they do tend to get a burst of transposition from a variety of different transposons. And so, we think that is the primary reason plants have acquired this sort of silencing process. It was not to confound the scientists that were going to investigate them, but rather to defend those plants against things that otherwise had no proteins associated with them, and therefore there was no way to really make a defense, an adequate defense. So I don't know if that's true, but it's the hypothesis that I thought fit the data they had. So let's just go back and review some of the details of silencing as we understand it now across the kingdoms, across the eukaryotic kingdoms. I want to point out, I'll mention one or two proteins here that I didn't talk about last time, but the main characters are still the same and the process is more or less the same. So, what we've learned from our previous studies was that double-stranded RNA, which is something that can be associated with virus replication, but can also be associated when we have large amounts of messenger coming from a particular integrated gene, can trigger the activation of Dicer. Dicer comes in and starts to cut the double-stranded RNA into 22 or 26 nucleotide chunks, and then other proteins come in, and one of them that I didn't talk about last time was called Argonaute, grabs a hold of these double-stranded sequences and transfers them to a multi-protein complex called a RISC complex. And it is that RISC complex that then dissociates the two strands and also triggers further rounds of dicing, and also potentially triggers site-specific DNA methylation as one of those strands is brought to the DNA to form a triple helix to attract DNA methylases, RNA-dependent DNA methylases. This drawing that I think I've got in your pictures there is an attempt to illustrate one such process. Now this, in a sense, is it's showing you two different types of materials. One is this double-stranded RNA here, and then the other would be this hairpin-like structure here. I mentioned very briefly that we have siRNAs, which are small RNAs that really result from the silencing process, and that we can even exploit to try to silence a gene of interest. But there's also another pathway that's somewhat related to these, that somewhat involves these enzymes, which involves the production of what are called microRNAs. There's not a heck of a lot of difference between a microRNA and an siRNA, except for the fact that we tend to be the agent responsible for siRNAs, and microRNAs are an integral part of gene regulation in the multicellular eukaryotes. So what they show in this drawing here is, regardless of whether you just have sort of double-stranded sequences like this or a hairpin structure where it folds back on itself, Dicer comes in, cuts them into small pieces, carries them to the RISC complex. The RISC complex then may deliver one of the antisense strands to the messenger RNA that is meant to be regulated, and that can then become a target for further dicing of the messenger, or it can become a primer for RDRP mediated extension of that small RNA so that we double-stranded RNA covers the entire length of the messenger. Then Dicer comes in and chops that double-stranded messenger into small pieces. In either case, we are destroying the messenger and increasing the number of copies of small RNAs to dice further messengers or to lead to the inhibition of further messengers. Now, in the case of these microRNAs that are being used in gene regulation, we don't actually want to destroy the RNA that we are targeting. The cell doesn't want to destroy the RNA that's being targeted. And so generally, microRNAs don't perfectly match the template they bind to. And because of that, they are not suitable substrates for further dicing. But they are capable of inhibiting, they are capable of two things. One, they're capable of being extended by RDRP, and two, they're capable of inhibiting the translation of that messenger if the microRNA is binding to the 5' end of the messenger. And in that way, when the cell says, look, I've got these regulatory proteins that are controlling the amount of messenger that's being produced, but then that messenger is getting grabbed by ribosomes and translated, and there's going to be a lag time in my ability as a cell to respond to overproduction of that protein. And so we're going to impose a second level of regulation where we don't destroy the excessive amount of messenger, we just inhibit its use. And so we see this in many, many genes within the cell, that they have both transcriptional and post-transcriptional controls imposed upon. Okay? Everybody all right there? Okay, maybe I just go on then. So at some point after these researchers, these people who are working in Beijing University, when these people introduced the two RDRPs, for whatever the reason, into the cells they happen to use are the cells that are most convenient for every mammalian biologist, which are rapidly growing, immortalized, cancer cells, effectively, oncocells. There's lots of cell cultures. You're all familiar probably with HeLa cells, but there's a lot of other more recent types of cell lines that have been generated. Now, anybody know why there's any advantage to studying these cancer cells as a basic model for mammalian gene studies? Why not use regular cells, non-tumorous cells? Okay.
Is it because they don't stop growing?
Yeah, they don't stop growing, whereas a normal cell line has a finite lifespan. You may have heard of Hayflick limit. Basically, a normal vertebrate cell can only undergo a certain number of divisions, and that number of divisions correlates with the actual average lifespan of the organism that they came from. So we do think in certain ways that whatever causes aging, it is dependent upon, or it's imprinted in the cells themselves that they can only divide so many times and then they stop. And that's one of the places where having a cell transformed into a cancer cell becomes problematic. Those cells don't know when to stop. They just keep proliferating and proliferating and proliferating. And so where they originated forms a mass of cells. And then sometimes under other conditions, those cells can detach and migrate to other locations in the body and start new colonies of these immortal cells. And in the end, suck up all the food and oxygen that a normal cell would need. But there are other reasons that really working with cancer cells makes life so much easier for a molecular biologist. And one of them is normal cells show a number of phenomena such as attachment-dependent growth, meaning they have to bind to a proper substrate made of extracellular matrix, if you know what that is, things like collagen, for example, in order for those cells to remain alive. If they don't bind to the proper proteins, which we then spread on the Petri dishes to make sure that they stay there, if they can't bind to that, they kill themselves because the cells figure, oh, I'm in the wrong location and I'm at risk of causing a disease, a cancer for the whole organism that I'm part of. And so they sacrifice themselves in order to prevent any damage from occurring. But that means when we grow normal mammalian cells, we have to culture them in dishes, and they have to be able to attach. We can't grow them in shakers like you would a bacterial or a yeast culture. And then an additional feature that normal cells display is what we call contact inhibition, which means that if they settle on a Petri dish, they're going to keep dividing until they all touch each other. And at that point, a signal is sent to the nucleus of each of those cells telling them to stop dividing. And again, this is a fail-safe mechanism to prevent things from overgrowing the organs that they are a part of. But in a cancer, that's another one of the pathways that has to get knocked out. So while a normal cell population would form a monolayer of cells on the petri dishes, to which we've already put extracellular matrix, cancer cells don't need to attach, and they don't care how concentrated they have become, whether they're touching each other or not. And as a result, we can culture these cancer cells much more easily, almost like bacteria, by having them shaking in a gentle shaking, nutrient bath. Okay? So there's lots of good reasons to use these cells. So, but what the strange things that happened were that when they transformed these cells with their genes, the cells stopped displaying the characteristics of a tumor. They stopped showing rapid migration. They seemed to show a need to settle down and attach to a substratum. And they looked at a number of criteria associated with that distinguish tumor cells from non-tumor cells, and almost every one of those showed a reversion from the tumorous condition to the more healthy condition, the condition we associate with a normal cell line. Obviously, if you saw something like this, you'd say this is really strange, and so I think in my mind, they changed the entire direction of their research and began to study how these proteins could actually reverse this type of cancer. So far, so good? So let's go through a little bit of background information. Again, some of you probably know this stuff, but it's just a review to show so that we have a common terminology. Whether a cell is growing normally or as a cancerous cell would grow is going to depend on how it's progressing through the cell cycle. Cell cycle is a term we use to describe the sequence of morphological and biochemical changes that occur in a cell from the time that it is first, split off of the mother cell to the time that it in turn splits into two cells. And as microscopists and later molecular biologists studied the order of events, they saw that, in fact, we could divide the cell cycle into 5 parts, or maybe 6, depending on how you define things. So each of these parts shows characteristic changes in the types of proteins that are being expressed. And each is a set unto itself, basically. You have to complete the production and the actions of the first set of proteins in order to initiate the actions of the second set of proteins. And then in order to initiate the actions of the third set, the fourth set, and the fifth set. So if we think of everything beginning at mitosis, then, let's see, do they have that here? Yeah, so here they're calling it cell division. Cytokinesis is another term you'll see. We start with this 2 freshly born mammalian cells, or any kind of eukaryotic cell. For a long period of time after division, we don't see much in the way of morphological changes. All these cells have an intact nucleus, an intact cell membrane. They're always making, at all times in their lives, they're all making proteins essential for translation, transcription, replication, not replication so much, but transcription, translation, and ability to respond to external stimuli and all of those things. So those are proteins that are made and can be made at any time in the cell cycle. But during this period, from the end of mitosis forward, we have no visible changes until after some period of time, all of a sudden, we begin to see DNA synthesis beginning, the synthesis of histones beginning, the synthesis of a number of other proteins associated with the nucleus and so on beginning. That's a visible thing that the microscopist could easily see even before we had tools to study things at the molecular level. So they decided to make that sort of the signpost for everything else other than mitosis that we see in the cell. They called that period S phase because they could show that during S phase, DNA in the nucleus is synthesized. No DNA is synthesized before that, and no DNA is essentially, except for repair processes, is synthesized after that. Well, then what do you call that time between the mitosis and the beginning of S? They didn't know what to call it, so they just called it a gap. And they called it G1, gap 1. Okay? We now know exactly what's going on in there, but it wasn't making any visible imprint on the cells themselves that the microscopist could see. Now, during S, all the DNA has to get doubled. All the DNA of the nucleus has to get doubled. And once that's been completed, DNA synthesis stops, and we have a, we have a new gap period. So that came to be called G2. Again, we don't see very much in the way of morphological changes in that time. But at the end of G2, we begin to see the synthesis of things like tubulin and all sorts of cell membrane proteins, cell wall proteins, and so on associated with cell division cytokinesis. And so after G2, we then have this period again of mitosis. And when mitosis is done, we go back into a new G1, and then a new S, and then a new G2, and then another mitosis. And the process repeats itself over and over again, so it's called a cycle. The ends and the beginnings of each of these phases is under very, very tight control. Now, if you think about it, everything that's going on it has the purpose of trying to replicate the mother cell, duplicate everything that's in the mother cell so that each of the daughters will start with the same amount of DNA, same genome essentially, same numbers of ribosomes, same proteins and other proteins and lipids and everything else, mitochondria, everything that the cell needs to, that those two daughter cells that will form will need to survive. So there are what we call checkpoint proteins that monitor progress through each of these phases. If something is wrong in G1, there will be a checkpoint protein that tells the cell to stop until those errors or those delays have been corrected. And then we go into S. Now S, we can easily imagine what can go wrong there. If the cell tries to divide too early, if S ends too early, what's going to happen is there's not going to be complete copies of each chromosome. And so when the cell divides, we're going to get aneuploid cells in the end. We also know DNA synthesis is full of errors. They're not at a high frequency, but we know that they do occur. On the other hand, the cell has proteins designed to correct those errors. And we need time for those proteins to find the mistakes and then correct them, because if they are not corrected, they lead to, what's the word? What do errors in DNA synthesis cause? Huh? Exactly. They will be mutations that will be passed on and uncorrectable from that point forward. So we need to give the cells time to correct those mistakes. Now we also use G2 for that same purpose. Again, there's going to be checkpoint proteins that are going to say, is S finished or not finished? Because if it's not finished, we can run into all sorts of problems. And then if S is finished, G2 is there's going to be a checkpoint protein in G2 that's going to investigate whether or not all of these, or at least most of these errors have been corrected. And if they haven't, it's going to stop progress through the cell cycle until that has occurred. And so, and then there'll be another kind of a checkpoint in mitosis. And so, each phase of the cell cycle represents a time when specific actions have to occur, and only at the completion of those actions, the successful completion of those actions, does the cell allow its biochemistry to move forward to the next phase. So far, so good? Well, cancer disrupts all of that stuff. And cancer is a pretty vague term. There are many, many different kinds of cancer, depending on which genes have been mutated by mistake. We have lots of genes that are involved in the cell cycle. But if some of those genes are mutated in such a way, not that they're inactive, but they're insensitive to the signals they're supposed to be recognizing, then that cell can continue to replicate even when it should not. Even when it is starved for nucleotides and therefore making lots of mistakes in the nucleus, it can't stop because the checkpoint proteins that should have stopped it at G1 have lost their activity through a mutation. And the same thing goes for all of the other properties that we think of when we look at cells. For a cancer to arise, we not only have to eliminate some of these checkpoints, we also have to eliminate the process that leads to suicidal cell death when cells detach from their substratum. We also need to eliminate that process that stops cell division when the cells contact the surface of other cells, and so on. And so no single mutation has been shown to ever give rise to a cancer. Every cancer has mutations in at least 3, if not 4, and sometimes many more genes in order to become tumorous. So in a sense, every cancer is quite different from every other cancer. And that's why this paper is so weird. Because what they've done is they have transformed, when they began to see things were strange with the one cell line they were using, the cancer cell line they were using, they got a hold of a few additional cancer cell lines and a normal I keep using the word untransformed, so just for people to understand why I'm messing up like that. In the old days, people said that tumor cells were transformed cells. And you'll still see that terminology in extensive use if you ever walk into a mammalian lab. Problem is that we have all semester been using transformed to indicate the introduction of foreign DNA into the cell. But when the cancer people are talking, all they're talking about is the cell has changed, transformed, in other words, from a normal cell to a cancer cell. So I don't want to let you get confused by connecting this term transformation with the way we've been using it so far. Okay? So just give me a little patience there, and every time I hiccup and start saying transform, I'm really going to try to correct myself and say cancerous, tumorous, or something like that, because that'll reduce the problem. Anyway, so they took all of these cells. They took several, so they did the first experiment with one cell line, and then they said, let's test some additional cancers. And what they did initially was they cloned the RDRP proteins into a lentivirus. What I said here? Did I say that? Well, into a virus that is simply like an RNA virus. It replicates, but it doesn't integrate. So in other words, what they would be doing is allowing for transient expression of the proteins they were looking at. This is a very common approach. These experiments take a lot of effort and a lot of time. You don't want to jump into getting stable transformants if you don't need them. So in order to test out, to do a preliminary set of experiments to see if this process is worth studying, they used a transient expression system that would transiently express these RDRP type proteins. And what they found was each of the cell lines they created or tested in this way showed the same results. So let's see if I can. Okay. So we're looking at a lung carcinoma, a liver carcinoma, another lung carcinoma, and a lymphoblast leukemia cell line. You can't see the colors here, but on your copy I hope you can see the colors. And what you're looking at here is that upper curve, parabola. There's actually two lines there overlapping each other. One of them, and what they're doing is they're just showing cell growth, cell division, the culture, the proliferation of the culture of a vector that's been transformed with the vector alone, one that's been transformed with an RDRP, one that's been transformed with a mutant RDRP that is inactive, functionally inactive. So they really tried a lot of controls here because I'm sure they hit a lot of criticism in the review process. And then what you see is, regardless if you have transformed these cells with a vector or you transform these cells with a non-functional RDRP, is all these cancers just keep proliferating and ad infinitum. On the other hand, if they transform them with a vector carrying either the Arabidopsis RDRP or the rice RDRP, AT means Arabidopsis, OS means rice, the proliferative state was greatly reduced. The cells did not show this very rapid, basically out of control cell proliferation. So this seemed to indicate that these RDRPs, regardless of their origin, whether they came from rice or Arabidopsis, had the same effect on multiple types of cancer. Now, we don't know exactly what the genes are that led to those cell lines becoming cancerous, but we can pretty much guess there weren't too many genes in common, too many mutations in common. So in theory, these RDRPs are inhibiting multiple causes of cancer. Well, they wanted to do a much more detailed study, so the next thing they did is they cloned the RDRP behind a tetracycline repressible promoter. We now know what a tetracycline repressible promoter is. This is a case where the surrounding the TATA box, we've placed some TET-O sequences so that when the TET repressor is co-expressed, it binds there and stops transcription. To start transcription of that gene, we add tetracycline. The tetracycline binds to the TET repressor, it changes its shape, it falls off the DNA, and now transcription can progress. In this particular case, as probably all cases today, they did actually not use tetracycline as the inducing molecule. They used a derivative that's called doxycycline, which is about 1,000 times more, has about 1,000 times higher affinity for the repressor. So it means you can use even less than you would a tetracycline. And it also has been shown not to have any biological activity other than that. So it doesn't harm the cell in any way. Tetracycline doesn't, but still, this is even better because of its higher affinity for the repressor. And so they cloned their RDRP behind that system so they could turn this on and off at will. And then they integrated, they cloned that chimeric construct now into what's called an AAV vector. AAV means adeno-associated virus. It's a small virus. It's perfectly harmless to everybody, humans and other mammals. And it can be used, it integrates into the genome, and it can be used to carry genes into the genome. Its only problem is that it can only, you can only put about 2.6 kb of foreign DNA in it. It's very it's very restricted in that way. But people have figured out ways to get around it. And so it's a pretty standard vector to be used for viral-mediated treatments, we hope, in the future. As far as I know, nobody has ever shown any kind of immunological reaction to AAV. Another virus that's commonly been used is adenovirus itself, and that has, in a few rare cases, triggered some immune reactions that have been lethal for the patients that were being tested on. So AAV, despite its limitations as far as cloning, seems to be the preferred method for putting new genes stably into the genome. So they cloned it in there, and they did a series of tests. And first of all, on its own, nothing happened. When they treated the cell lines with doxycycline, they began to lose their cancerous characteristics, presumably because of the production of the RDRP. And they did this for a whole variety of cell lines. In a review I was just looking at before coming to class, they've extended that to at least 30 independently derived cancer lines. So think about it for a moment. This is a weird thing that this one treatment seems to be almost universal with regards to cancer. We have nothing else like this. We have some drugs that will inhibit some key cancer genes, very common genes, but we don't have, but each cancer needs to be treated for the defects in multiple genes. And here it looks like we're getting something that affects all cancers regardless of how they were instigated. And what we're looking at here, which I didn't put into your thing, I realized afterwards that I needed to show you this, are Western analyses of a series of proteins. So what they had done when they got this result is they did somewhat like a transcriptome or a proteomic analysis. And what they found is that while the genes in a natural cell line, a normal healthy cell line, don't show changes when they're infected with this RDRP, in the cancer cell lines they saw changes in about at least 30 different cell cycle genes. And here what they're looking at is they're showing you what happens when we turn RDRP on to some of the key kinds of genes that we think about when we're talking about cancer. So beta-actin is our control. This is a gene. This is a common housekeeping gene. It's found in all cell lines. Don't actually remember. I think it's phospholipase K. I'm not sure actually now. Now I can't think of what this one is. MCM2 is one of those checkpoint proteins, however. CDK6 is a kinase that's responsible for controlling parts of the cell cycle. CCNE is a subunit of the kinase that controls the cell cycle. It's called cyclin E, critical for starting the cell cycle at the beginning of every cycle. And then the other one is cyclin D, which is the second cyclin that's really critical for starting the cell cycle. So we're looking at really important kinds of proteins. Now, this cell line is a natural cell line. It is not cancerous, okay? And what you see here is when they are looking at the levels of these proteins, either with the doxycycline or without the doxycycline, with the doxycycline or without the doxycycline, so these two are replicates without, these two are replicates with, we don't see significant changes in the expression of any of those genes. This is 3T3 cells. This is an embryogenic cell line in common use. This also doesn't show very much, although they did notice that with cyclin E there may have been some slight reduction in cyclin E, one of the key proteins in starting the cell cycle. when we treated those cells with doxycycline. But if you look at these cell lines, these are cancer cell lines. What you see here is though the actin doesn't change with or without the doxycycline, when you had doxycycline, for example, let's look at the ones I know about, the MCM2 levels drop. And they didn't drop in the normal cell line. The CDK6 levels drop. The levels of cyclin E drop, and the levels of cyclin D drop. And so, and then this process is the same in this cell line and in the HeLa cells, the infamous HeLa cells. So what the idea here is that something has gone wrong in the regulation of these proteins in these cancer cell lines, so that the cell is just unable to completely shut them off. And as a result, we are getting all the traits of a cancer cell. When we put the RDRP in there, we seem to restore inhibition of these kinds of proteins. These proteins should only be produced, well, especially these ones up here, these three up here, should only be produced at the beginning of the cell cycle, not other times. And so normally they should be in rather low amounts in these cell cultures, but they weren't. And what you're seeing here is that we can reduce those concentrations, not completely, but by maybe 50, 60, or 70%, depending on the protein, and a little bit depending on the replication of the experiment. Okay, so everybody so far so good there? Now, when they did a more in-depth analysis of these kinds of cells, they saw that there was something wrong with the production of certain microRNAs. The microRNAs that normally would be down-regulating those very cell cycle proteins. This is again just sort of summarizing what we think is going on with these microRNAs. We think that the microRNAs generally bind, like in the ones that we're showing here, normally bind to the 3' end of a messenger, and then they lead to dicing of the messenger. They all have these sorts of, that little blip there is intended to show that these microRNAs, unlike an siRNA, doesn't bind perfectly to its target. So it doesn't form a stable enough complex to be captured. So they started to isolate these these microRNAs that we're talking about, as well as some siRNAs, and that would result from the destruction of those cell cycle proteins. Normally, and here's things getting a little weird. So normally, dicer from plants has, in addition to its cleavage ability, also has an additional activity which is called a nucleotidal transferase activity. This is the ability of the enzyme to attach a single nucleotide to the 3' end of these small RNAs. In mammals, we don't have a functioning RDRP protein, but they do have another enzyme, whose name I can't recall for the moment, nucleotidal transferase, that basically would do the same function, but it wouldn't have dicer activity. But the point is that for these double-stranded RNAs to be captured by the RISC complex, they have to have a 2-nucleotide overhang on the top strand and on the bottom strand. Think of it like a sticky end, two nucleotide sticky end. If they don't have it, they are not bound tightly enough by the RISC complex to be utilized efficiently. And what they saw is in 30 to 70% of the cell lines that they looked at, the cancer cell lines they looked at, the small microRNAs and even the siRNAs that were accumulating only had a single base overhang, and theoretically therefore could not function, could not be captured easily. Knowing then that the dicer itself, the plant dicer, would be able to add that nucleotide, they then did some serious acrylamide gel analyses, and well, this let me show you this point first. So this is actually a demonstration of the abnormal size of these SI or microRNAs in cancer cell lines. So here we have, again, this RPE is the normal cell line, and we're looking at several different microRNAs. And what you see is that in this cell line, this natural cell line, there was only one size class for that microRNA. So you see only this one band here. But in each of these cancer cell lines, these microRNAs that are shown here, here, and here, which are intended to bind to some of the cell cycle genes to downregulate their expression, you see two bands separated with one nucleotide difference between them. So it's as if something has gone wrong in the nucleotide processing of these cancer cell lines, something nobody had guessed at before, leading to incomplete tailing, if you will, of the two RNA strands. And what they then found when they put the RDRP into these cells is they reduced that bottom band. Yeah. They reduced that bottom band so that most, if not all, of the microRNAs produced had the proper two nucleotide overhang, sticky ends, if you like. Now, they don't think any part of any other activity that the RDRP can carry out is important here. They think this is probably the only feature that really matters as far as things go. They don't know why cancer cells seem to, regardless of their origin, seem to only have a single nucleotide sticky end. So they don't know that, but what they can say is that this protein cures that problem. So if we look at it in a summary form here, in normal cells, microRNAs, and so these are the RNAs that are going to be downregulating, in our case, a number of cell cycle genes, such as the cyclin E and CD36, and another cell cycle protein, this is a phosphatase, it's called CBC25. That these proteins are only made in enough, the amounts that the cell actually needs, and no excess is produced. These microRNAs initially are transcribed as something that can form a long hairpin, and then a protein that's called here drosha. This is another protein like dicer, but not exactly like dicer, but I haven't talked about it because it's only involved in microRNA processing. Cuts these guys up, and then those pieces then with their little two base overhangs are carried to RISC, and they bind there and inhibit the translation of those proteins so that we fine-tune the amounts of these critical cell cycle genes. But for some reason that we don't understand, in cancer cells, something is removing one of the two overhanging bases, giving us only a one nucleotide overhang. And this leads to improper or weak binding to the RISC complex, and this leads to the inability of these microRNAs to downregulate the production of the cell cycle proteins. When we put RDRP in there, we reverse all of this. It cures, literally cures, as it's done in these mice. So they're not only working in cell lines, they're also working in cell lines they've injected into mice to form tumors. Cell lines transformed with these RDRPs lose their cancer properties. It's shocking, and we'll have to wait and see, you know, if these guys have done it right. But if this is true, there's some really great work for anybody out there in the class who's interested in doing kind of work like this. We want to know why these cancer cells don't have that two-base overhang. We want to know more about how the regulation of all these genes is being maintained. There's just so many things. Why is it so universal? I mean, for me, that's the biggest question. Why does it seem to be universal? Is it truly universal? Like I said, they've tested 30 cell lines now, and it seems to be universal. We've just never had a single target to aim for when we wanted to find drugs to treat cancer. If we can figure out what's going on here, we might actually be able to find a single drug that could have this effect. We're probably never going to be injecting RDRPs into people. But if we can find the protein that's going wrong here, we might find a way to change that protein so that the microRNAs are processed correctly with the two-base overhangs. All right, that's all. That's all I was going to show you. Is there any questions on any of this? It's okay. If not, does anybody have any questions on those things that I handed out? Yeah.
So I have multiple questions, and I kind of have them, I guess, organized. So one question that I just genuinely did not know what you were asking. Maybe I'm tired and reading it wrong. The question is why do we often associate silencing with transformants where the transgene integrated 3' end to 3' end? And I guess I just don't understand what that question is asking.
Let me think when I talked about this. Okay, if you might recall, at the end of the last lecture, I had a piece of paper with 4 drawings on it, intended to show things that could cause silencing. One of them was, overproduction of a retrotransposon. I'm not even sure I remember what the others were, but one of them, the second one, if I recall, showed a drawing with an inverted repeat of the integrated gene. So what happens is when you put DNA into a cell, some of it is going to integrate. It doesn't, in many cases, it integrates, each molecule that's integrating goes to a different location. In some cases, if you've limited the number of molecules that you inject, you may only get one molecule integrating, which is the ideal situation. But in certain cases, when you're putting in thousands of copies of the same gene, you can get multiple integrations. And sometimes, because they're all going into this, these nicks, you may even get copies in the same spot. There's also a process that's been identified in plants, and I don't know if it's been identified in mammals, where the integrated DNA undergoes some kind of a replicative process, even before it integrates. The introduced DNA undergoes some kind of replicative process. And what happens is basically that DNA polymerase copies the top strands, and then instead of falling off, as it should, it just switches templates and goes to the bottom strand, and the result is the formation of a long inverted repeat copy of the DNA that you put in there. Then that becomes double-stranded. And so now we have tail-to-tail, a tail-to-tail construct of the DNA we first introduced. And then some of those integrate, and now we have a chromosome that has inverted repeats in it, made up of the gene we put in. Can you people picture that? Is that okay? So, I mean, the mechanism is not so critical, but what matters is that you got an inverted repeat. And the reason that's important is because that means each end had a promoter, so transcription could go on opposite strands from each promoter. And although it should have terminated at the terminator, you know that we really don't have a good terminator for eukaryotes. So yeah, there's some chances for termination, but there's also chances that the polymerases starting at the ends would just keep going and copy the entire inverted sequence. And when that happens, the RNA now consists of an inverted repeat of the transgene. It can fold over, and now we have a stem and loop structure that looks just like these microRNAs that I've shown you, or like everything else that triggers silencing. So my point in asking this question was for you to kind of retell that story for me.
Okay. I think I was confused by like the wording of 3' end to 3' end integration. I was thinking about like the term inverted repeat was the phrase that I needed to hear in regards to that.
And maybe I should have said that, but I thought that, I don't know, I guess I wanted to stretch your imagination.
No, that's fair. I also looked at this when I was very tired, so it's entirely possible I just
Sure.
didn't.
Well, I know that.
Yeah.
I was writing it when I was tired. Okay. Why don't we just see if anybody else has a question while you're selecting your next one. Anybody have a question now? Okay. Do you have another question?
I have many other questions.
Okay, well, you're doing a good job for the class.
These two questions, they kind of go together. And going through my notes, again, I may have just missed something, and like not written it down or not been able to find it. But there's two questions. One of them is, in what ways are the methylation processes in bacteria like those in eukaryotes? And the other one is, how are they different? And the only thing that I could really think of was like euchromatin versus heterochromatin, or yeah, those. Sorry, again, tired. Was like the way that chromatin is organized in eukaryotes, but I wasn't entirely sure if that was in the right direction or not.
All right. I probably will, maybe I need to think a little bit more about the question. But I really was intending people to focus on what, I mean, actually, I guess I did ask that they for a purpose. You could have answered this in a number of ways, including you could have talked to me about the mechanisms of, you know, if we were to talk about what is a common feature between methylation in a bacterium and methylation in a eukaryote? What would you guess is a common feature?
Well, I assumed that the actual like process was the same, but I
And
could
what
be
would be
very
that
wrong
process?
about that. Where like a DNA methyltransferase comes in and interacts with a cytosine.
Okay, and that's the key word.
Okay.
In both cases, the predominant form of methylation is on cytosines. Now there is amethylation, but we don't care about it because it's got limited value in eukaryotes. In bacteria, it actually is, amethylation is one of the regulators for the cell cycle. But we don't care about that. Okay, so if we asked for what was similar, that could be a similarity. Now, in terms of differences, anybody want to tell me what is a difference? Think functionally, because everything in the course here is intended to ask you to think about function more than structure. So think functionally. What is the purpose of methylation in a bacteria? Okay, Faith?
Well, when I think of bacteria, I think of restriction enzymes when it comes to methylation
Right.
and resistance to those
And resistance
restriction
to those.
enzymes. And when I think about eukaryotes, I think about more gene regulation.
Right. So there are no restriction enzymes in multicellular eukaryotes. So that's a huge difference. In bacteria, it protects the DNA from restriction. In eukaryotes, it modulates the expression of the genes. Inhibits the expression. I shouldn't even say modulates. It purposefully, truly inhibits the methylation of the genes. I mean the transcription of the genes. So for me, that would be a desirable answer. Okay. Any other questions?
I do have more if no one else has any others.
I just wanted to check. Yeah, just for the people that are going to be watching this video, as I wrote in the email, if anybody not in the classroom has a question on Wednesday, write to me by email. And I'll have my computer here, and I'll try to answer those questions in addition to whatever comes out of class. But if there's no email questions and there's no more questions in class, we're going to have a very short meeting on Wednesday, which I know you'd appreciate anyway. Do we have any other burning questions, or we can just continue on Wednesday if you want? Do you have a burning question? I know you have a lot.
I do have a lot of questions.
Well, go ahead and try one more. Let's see how it goes.
Okay. Let's see. Oh, one question that I had is a question that's asking, would an enhancer work if inserted into 3 different regions? One is an intron, one is after the site where the poly A tail is added, and one is at the one is overlapping the ORF. And so I know that we've said that enhancers are position independent. However, when I was looking at this question, I realized that we never talked about if it's actually in the gene. So I was confused about that part.
Yeah, so I mean, technically it's position independent.
Mm-hmm.
You can put it anywhere, including in the coding region.
Okay.
There are a number of genes where it's in an intron, but there's no reason that you could not have an enhancer in the coding region other than the fact that it would be a protein there that might block the transcription of that region. But technically, from the perspective of functionality, it would still trigger transcription. And when people first discovered these enhancers, you know, when they were studying a virus called SV40, a mammalian virus called SV40, they demanded that everybody who thought they had an enhancer clone the enhancer at the 3' end of the gene past the poly A addition site. And if it didn't work there, it weren't no enhancer that they ever heard of before. But every one that they tested actually worked, no matter whether it was at the 5' upstream of the promoter or downstream from the poly A sequence or put into an intron. All right. Well, why don't we just pause then? And there's time to look over those things and to come back with new questions on Wednesday. I do want to mention by the by that I'm going to have a number of extra credit questions on the exam that don't appear on those study sheets, although they are related to the questions on those study sheets. They're really for people who have tried to, go beyond the guidelines that I've given you. I mean, I'm not going to be asking you extra credit questions on topics I've never discussed, and I'm not going to be asking you extra credit questions on what at least I consider as picayune, minor, trivial details, They're going to be basic questions, but they're going to be questions that for one reason or another they may have appeared in previous sets of focus questions, but they just, for one reason or another, didn't appear in this recent set. So if you do have the chance to study beyond, the allotted number of questions that I gave you, you may actually be able to earn some extra credit, and who knows, that could be very beneficial for some people. Okay? So, just for those who want to overachieve. Okay, we'll stop. I hope I see you Wednesday, but I understand it if you don't want to come.